Hello there guys, and welcome to the Biology Mind A-level tutorial on transport across membranes. And in this tutorial we're going to have a look at diffusion in particular. So just to recap, in the last lesson we learned about the structure of cell membranes and how the cell surface membranes and membranes around the cell organelles of eukaryotes are the same. Now today we have 4 key learning objectives. So number 1, just having a look at the definition of simple diffusion. Number 2, having a look at some factors affecting simple diffusion. Number 3, having a look at facilitated diffusion. And number 4, having a look at factors affecting facilitated diffusion. So let's crack on and kick off with what simple diffusion is. Now, simple diffusion is net movement of particles down a concentration gradient from a high concentration to a low concentration. So this is a definition that you need to commit to memory. So I'll just put this here to remind you. And let's just break it down and think about what it actually means. So let's start by having a look at this concentration gradient thing. So let's have a look at this beaker that we have. This is our semi-permeable membrane. This is our high concentration. High concentration, I'll just write high conc, and this is our low concentration. And what we're getting with diffusion is we're getting our movement of particles down a concentration gradient from a high concentration gradient over here to a low concentration gradient. So if you give it a bit of time, we're going to get an equalisation of the concentrations on either side of our semi-permeable membrane. So we've covered most of what the definition is, apart from this meaning of net. Now, what we mean by net is the overall trend of movement of the particles is down the concentration gradient. But what you have to remember is various particles are moving in different directions from either low to high or high to low concentration, but overall most particles are moving from high to low. So that's why it's really important you include this word net, because that describes that the overall trend of the particles is to move down a concentration gradient, but it also acknowledges that some move back the other way, because remember our membrane is semi-permeable, you can move through it as much as you want. So diffusion occurs until dynamic equilibrium. So this is dynamic equilibrium. So i.e. where we have the same concentration of the substance on either side of the semi-permeable membrane. Okay, so diffusion is passive. It does not require energy. Now, what does this contrast to? So in GCSE you would have met active transport, which requires energy. So diffusion is passive, okay? Does not require energy. Now just a little tip from StudyMind, which is to learn the definition of diffusion word by word. So that definition we had on the first slide, learn it word by word, because it's really important to use words such as particles, because this encompasses molecules, ions and atoms. It doesn't exclude anything. It's also important to use the word net, as I banged on about earlier, because some particles move up the concentration gradient and some move down it, but most of them are moving down the concentration gradient, just like what our definition of diffusion says. Okay, so factors affecting the rate of simple diffusion. So again, these are just things that you have to learn. So number 1, the size of the concentration gradient. So the greater the difference in concentration between the two regions, the greater the concentration gradient. So the greater the concentration gradient, the faster the rate of simple diffusion. Number 2, diffusion distance. So rate is inversely proportional to the distance between the two regions. So therefore that means that over shorter distances the rate of diffusion is faster. So we've got to decrease the diffusion distance in order to favour our rate of diffusion. That's what the word inversely proportional means. Surface area is our number 3 factor, so the greater the surface area of the membrane, the more diffusion occurs. This makes sense because it allows a larger surface area for these particles to diffuse across the membrane, instead of like, a queue forming across a short membrane, for example. So then number 4 is our size of our molecule. So smaller molecules diffuse faster. Number 5 is our temperature. So remember, increasing temperature of a substance increases the kinetic energy of the particles, which increases the rate of diffusion. And then finally, number 6, stirring. This is applicable to experiments. So for example, if you give the beaker a stir, that's going to allow particles to move around faster, and it's going to allow the particles to diffuse through the membrane faster and more easily. Obviously, in your body, stirring does not occur, or in cells, stirring does not occur, so you wouldn't get this in a living substance. Okay, now just a point on membrane thickness is the membrane has to be very thin to provide the shortest path length for the substances to diffuse across. So this is our membrane. We want it to be nice and thin so that there isn't a huge distance for the substance to diffuse across. And in terms of the phospholipid bilayer, remember it has this barrier function, which means that it allows some substances to pass through and some substances to not pass through. So small non-polar molecules can simply diffuse through the phospholipid bilayer, just naturally through the bilayer. Now, just a tip on what's polar and what's not polar. So to generalise, ions are polar molecules and lipid-based molecules are non-polar. Now, very small molecules and ions, for example, carbon dioxide, are small enough to diffuse through the phospholipid bilayer. In facilitated diffusion, we have proteins within the phospholipid bilayer that we've met in the previous tutorial that allow for our polar molecules and ions to diffuse into and out the cell. So these are called carrier proteins, such as seen here, and channel proteins. So polar molecules cannot pass by simple diffusion, and that's because, remember, they're polar, they carry a charge, so they can't cross the hydrophobic barrier. So instead they have to travel through the carrier proteins, because the carrier proteins themselves allow for this to happen. So polar and hydrophilic molecules travel through carrier and channel proteins. They are facilitated across, or helped across. Now, let's have a look at why these proteins can allow for this to occur. So these proteins have pores that are filled with water to attract hydrophilic molecules. And the proteins in this region here are polar, which allows them to interact with other charged molecules, so other polar molecules, and allows them to travel through these pores to enter or exit a cell. Channel proteins are less selective and mildly discriminate between the molecules based on size and charge. Carrier proteins are more selective than channel proteins and only allow one specific type of molecule to cross. Okay, so let's just recap that. So we have our channel proteins, we have our carrier proteins that are allowing for facilitated diffusion. So now we're going to come on to think about what can affect the rate of facilitated diffusion. So number 1, simple diffusion factors, such as what we've described on the previous slide, the last table that we saw. So, things like temperature, things like thickness of the membrane, things like the concentration gradient. But as well as that, other things that affect the rate of facilitated diffusion include the number and size of transporters. So the more transport proteins present, the greater the rate of diffusion. If all the transport proteins are saturated, then increasing the concentration will have a minimal effect. So we need to increase the number of proteins that we have to carry our particles across. And number 3, regulation by cells. So increasing temperature increases kinetic energy, which increases the rate of diffusion. Okay, so that's our lesson for today complete. Well done on following through, and I'll see you for the next one.